Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International GCSE Biology Paper 1BR for January 2023. Let us begin with the first question. Question 1. Gas exchange occurs in the respiratory system. The human respiratory system is located in the thorax. In part A, the diagram shows some of the structures in the human thorax. So they say, what is the name of the structure labeled X? These are bronchioles, and therefore the answer should be B. Next they say, which of these describes what happens when a person breathes in? So they say the diaphragm contracts and moves downwards. That is correct. The diaphragm contracts and moves upwards. That is wrong. And these statements about the diaphragm relaxing are also wrong, so A is the only correct answer. Moving on. In part B, the table shows the percentage of some of the gases in inhaled air and exhaled air. So we can see the table with the information. We see there is oxygen, which is 20 here, and that is 16, meaning it decreased. Carbon dioxide increased, and nitrogen remained the same. They say explain the differences between the percentages of gases in inhaled air and in exhaled air. Remember, this statement is about explanation, so we have to state what differences there are, and then give a reason as to why those differences are the way they are. So I said, inhaled air contains more oxygen than exhaled air because oxygen is used during respiration in the cells. When air is exhaled, there is less oxygen because it has been used up. Then, exhaled air contains more carbon dioxide than inhaled air because carbon dioxide is produced during respiration. Both inhaled and exhaled air have the same percentage of nitrogen because cells do not use up or release nitrogen. Down here they say, air in the alveoli has a composition of 14% oxygen and 7% carbon dioxide. Suggest why the composition of air in the alveoli is different from the exhaled air. Remember, we're talking about the alveoli. They contain exhaled as well as air to be inhaled. So I say no gaseous exchange occurs in the trachea, so the alveoli contain both exhaled and alveolar air, so the composition is going to be different. So this brings us to the end of question 1. Let's continue to question 2. Question 2. Tobacco mosaic virus infects plant cells. The photograph shows some of the virus particles. So we can see the particles in here. They say tobacco mosaic virus particles consist of a molecule of RNA surrounded by a coat. Which substance is the coat made from? The coat is made from a protein. So the answer should be a C. Next they say, the virus particle labeled P has an actual length of 0.3 micrometer. Calculate the magnification of this virus particle. So they've given you 1 millimeter is 1,000 micrometer. Here you have to use your ruler and get the actual dimensions of particle P. It is about 2.4, 2.3 centimeters. When you convert that to millimeters, it's going to be 23 or 24 millimeters. But we know 1 millimeter is 1,000 micrometer. Therefore, this is going to be 2 3 times 1,000 micrometer. Magnification is going to be image size divided by object size. And I got this as my answer. Moving on. In part B, the photograph shows the leaves of a plant infected by tobacco mosaic virus. So we can see the plant there. They say plant cells infected by the virus stop making chlorophyll. Explain why plants that are infected with a virus grow more slowly than uninfected plants. Because they stop making chlorophyll, it means there is going to be less trapping of light, and therefore there is going to be less light absorbed for photosynthesis. So less photosynthesis will take place, less glucose is going to be made, and therefore there will be less respiration, so less ATP will be formed. But remember, ATP is vital in cellular processes like mitosis and so on. Also, glucose is required in the formation of starch as well as cellulose and amino acids, as well as other components within the plant. Even formation of lipids depends on glucose or depends on photosynthesis. So if those are not made, it means new cells cannot be formed and growth is going to go down or it's going to decrease. So there will be less mitosis and therefore there will be less growth. So this brings us to the end of question 2. Let's continue to question 3. Question 3, the diagram shows part of a woodland food web. So this is the food web. They say which organism is a primary consumer in this food web. Primary consumers are this, that, and that. So we look at the possible examples. There is an aphid, there is no caterpillar and no mouse, so I'm going to go with the aphid. Therefore, my answer is A. Next, they say, which of these describes the woodland community? A, all the abiotic and biotic factors in an area. No, this is talking about the ecosystem. Or the blackbird in the area. That is talking about a population because these are the same species. Or the different species in the habitat. That is about ecosystem. And then all the different species in an area. This is about community, and therefore this is going to be the answer. Moving on. In part B, this is one food chain from the food web. So we can see the oak, then the mouse and fox. Of course, the mouse is feeding on the oak tree. The fox is feeding on the mouse. They say a student counted 20 mice living around 4 oak trees. So this is 20. There were also 2 foxes, which is 2 in the area. So we have 4 oak trees, 20 mice, and 2 foxes. They say draw a labeled pyramid of numbers from this food chain on the grid. Your pyramid should be drawn to scale. Now, since I know these are going to be 4 and these are going to be 20 and that is going to be 2, I chose the middle part because they say to scale, so I try to position everything uniformly. So since this is going to be 20 and vertically they have to be the same, so I used the horizontal to give me the numbers. So for 20, I used 5 boxes. You can see 1, 2, 3, 4, and 5. And then for this one here, that is going to be 4. I used 1 box because, of course, 20 divided by 5 gives you 4. So if there are 5 boxes here, this is going to be 1. And it means the top one is going to be half of this. So meaning if these are 10, that is going to be 5 squares. But again, positioned in the middle, that's why you see this is half and that is half. So this is going to be the oak tree or oak trees. That is going to be the mice or mouse. And that is going to be the foxes that are just 2. Down here they say, photosynthesis results in 25,000 kilojoules of energy being transferred to the biomass of the oak tree each year. Only 8% of this energy is transferred to the biomass of the 20 mice each year. Calculate the mean amount of energy transferred to the biomass of 1 mouse each year. So 8% of this is transferred to all the 20, meaning 8 over 100 times 25,000 gives us that. Now for each mouse, I have to divide that by 20, giving me 100 kilojoules of energy that is transferred per mouse per year. Moving on. Here they say, explain why only 8% of the energy stored in the biomass of the oak trees is used by the mice for growth. This is quite easy. There is going to be energy losses as well as substances that are not eaten or that are not digested. So I said energy is lost as heat during respiration. Some parts of the oak trees are not eaten. Some parts like leaves can fall off and decompose before being eaten. Some eaten parts are not digested, so they are egested. And some parts are excreted in urine like urea as well as sweat. So this brings us to the end of question 3. Let's continue to question 4. Question 4. A cow's milk contains a mixture of nutrients, including protein, fat, sugar, vitamins, and minerals. The main sugar in milk is a carbohydrate called lactose. Which chemical elements are present in carbohydrates? Because they talked about elements, carbohydrates contain only carbon, hydrogen, and oxygen, so B should be the answer. Down here they say, describe how to test a sample of milk for protein. To test for protein, you're going to use butyrate solution, and if you observe a purple color, then that confirms presence of a protein. Down here they say, some people drink a milk alternative produced from plants, such as soy and rice, instead of milk produced by animals. The table shows nutritional information for different milk products. So here we see the products from cows, soy, as well as rice. When you compare the energy, there is more energy here. The least energy is here. This has intermediate amount of energy. It could be able to sustain life. Then when we go to mass of a protein, these have the same amount of protein, where this one contains a very low amount of protein. So taking rice products will deprive your proteins, and there is a chance of getting some food deficiency syndromes. Then mass of fat in milligrams. This one has a higher mass of fat. This one has half the amount of fat here. So this could be good for the body, not too much fat. But this is very low. It could be disadvantageous because some of these fats contain lipids that are beneficial as an energy source, or they could be beneficial in the formation of components within a cell. Then mass of carbohydrate in grams. This has quite high, which is good for energy, but if it's too much, then it's going to be stored as fat in the body, while this one has low. Then mass of calcium. This one has more mass of calcium, which is good because we need calcium for bones as well as teeth development. So lack of calcium is going to cause weak bones as well as deficiency diseases like rickets. So here they say the recommended daily amount of calcium for a child is 1,400 milligrams. Calculate the mass of cow's milk that would contain 1,400 milligrams of calcium. If you go back to the table, from the table 2 to 4 grams contain 250 milligrams. If I take you back here, 2 to 4 grams of milk contain that. So here we can say 1 milligram is contained in 2 to 4 divided by 250, and therefore this is going to contain that times 1,400, which is going to give us 1,254.4 grams. So that should be the answer. Next they say, discuss whether soy or rice milk is a suitable replacement for cow's milk in the diet of children. I want to take you back here, and again I've gone through this. When we see the comparison, soy has lower energy, it has the same amount of protein, which is good. So proteins are useful in the formation of components within the body as well as growth and repair. Here we can see fat is halfway, which could be good, but this one has very low fat, this one has lower mass of carbs, this one has higher, and this one has high calcium while this one has none. So in my answer I said, both soy and rice contain lower energy than in cow milk. Soy contains the lowest energy. Soy contains the same mass of protein as cow milk, but rice contains very low protein. This could lead to less growth as well as repair. Soy contains half the mass of fat contained in cow milk, but rice contains a very low mass of fat. Rice contains a higher mass of carbohydrates than in cow milk, while the mass of carbohydrates in soy is lower. So carbohydrates in soy could be enough. Soy contains more calcium than in cow milk, while rice has zero calcium. Lack of calcium can lead to rickets as well as weak teeth. Overall, soy is closer to cow milk, so it can be a suitable replacement. So this brings us to the end of question 4. Let's continue to question 5. Question 5, the photograph shows a red fire ant. This is the ant we're talking about. Fire ants are pests and damage crops. Forage flies can be used as a biological control of fire ants. Scientists use this method to investigate the use of pesticides and biological control on populations of fire ants that live on plants in glass houses. In one glass house, keep plants with no treatment. In a second glass house, treat plants with pesticide. In a third glass house, treat plants with pesticide and then immediately introduce forage flies. Count the number of fire ants in each glass house at intervals for 24 months. The table shows the scientists' results. So here from the table we can see time is increasing in months. This increases, decreases, and increases. And then here we can see there is a decrease until here, then it begins to increase. Here we can see a decrease and then it becomes constant. So they say give a reason why the scientists include results from a glass house with no treatment in their investigation. This is to ensure that any change that is observed in the study is solely due to the treatment and not any other conditions. So I said it is a control to check if the change is solely due to the treatment. Actually, even if you don't write this, just say it is a control experiment. They allow that. In part B, draw a line graph to show the changes in the number of fire ants with pesticides only and with both pesticide and forage flies for 24 months. So this is the number of ants, and again the information I used is from this table here. I'm taking this result as well as that result. So this is going to be the time on the horizontal axis, and this is going to be on the vertical axis as well as that. And for each, we're going to have line graphs. Please keep in mind that these are the same and that are the same. So those locations are going to be at the same point, as you can see here as well as here. So ranging from 0 to 500 and then from 0 to 24 months, make sure you use more than half of the grid provided. So this is more than a half. I position my points, as you can see, for the first one and then for the second one down here. So here they say, describe the effect of using pesticides only on the number of fire ants for 24 months. when we use pesticides only, the number of ants decrease for the first 6 months and then they begin to increase. So that is my answer here. Moving on. In part A, discuss the effect of using pesticides only and of using both pesticides and phorid flies. I want you to use data and your own biological knowledge in your answer. So what I wrote here is something I've already explained. If you can see here, when we use a mixture of pesticides and phorid flies, it decreases greatly for the first 6 months, then a slow decrease and then remains constant. Well, here we can see there is a decrease and then an increase. So here I said, with no treatment, the ants increase in number. This is from the table. Then both treatments resulted in a decrease in the number of ants for the first 6 months. With pesticide only, the number of red fire ants increased after the first 6 months, while when pesticides and phorid flies are used, the number of ants decreases and becomes constant at 12 months. The number of ants is constant because the rate of reproduction is equal to the rate of predation. So phorid flies act as a biological control in this experiment. So this brings us to the end of question 5. Let's continue to question 6. Question 6. The following passage is about chemical coordination in animal and plants. Complete the passage by writing a suitable word in the blank space. Animals and plants use chemicals to coordinate responses. In animals, some glands produce hormones which are transported in the plasma of blood, because this is plasma within the blood. A higher glucose concentration in the blood stimulates the release of a hormone called insulin. This is due to higher concentration of glucose from its release from the pancreas, and this causes the organ called the liver, or you could say the muscles, to remove glucose from the blood and store it as a substance called glycogen. Then plant shoots grow towards light. This response is called phototropism, or you could say positive phototropism, and the movement of a chemical called auxin to the shaded side of the shoot causes the shoot to grow towards the light. So this brings us to the end of question 6. Let's continue to question 7. Question 7. A species of grass has a diploid chromosome number of 12. Which row in the table shows the number of chromosomes in the nucleus of a pollen grain and the nucleus of a root cell of this grass? So remember, they said a diploid is 12, so the haploid has to be 6. Since the pollen grains are haploid, the answer for this part should be 6, and the roots are tissues of the plant, so they're going to be diploid, so the answer is going to be B here. Moving on. In part B, the diagram shows a flower from the grass plant. So we have the stigma as well as the anthers. They say explain 2 ways this flower is adapted for wind pollination. The stigma is feathery so that it can trap the pollen, and then we can see the anthers are exposed so that the wind can be able to blow the pollen away from them in order for it to be dispersed. Moving on. In part C, scientists use this method to investigate self-pollination and cross-pollination in a species of grass plant. Take pollen grains from the anther of a flower, then place some of the pollen grains on the stigma of the same flower. After 1 day, count how many of the pollen grains grow a pollen tube. Then repeat the method, but place the pollen grains on the stigma of a flower of a different plant. The table shows the scientists' results. So we see pollen transfer to the stigma of the same plant. The percentage growth of the pollen tube is going to be 5%. And then pollen transfer to the stigma of a flower on a different plant, we see 75% growth. So they say state the dependent variable in this investigation. The dependent variable is the measured variable, so I said the number of pollen tubes that are grown or that germinate. The next here they say, explain how the results of this investigation show that this species of grass plant is able to survive in a changing environment. Use the information in the table to support your answer. So in this case, we saw that fewer pollen grains were able to grow when placed on the stigma of the same plant, while on the stigma of a different plant, more grew. It means more cross-pollination occurs than self-pollination. And if cross-pollination occurs, there is going to be an increase in the genetic variation, and that means the plants are going to have better adaptation in order to survive when conditions change. So I said, fewer pollen grains form pollen tubes with self-pollination, yet more pollen grains form pollen tubes with cross-pollination. Cross-pollination increases genetic variation, So when a selection pressure arises, some plants will have a selective advantage to adapt to the change. Moving on. In part D, grass plants can also be produced by asexual reproduction. New plants are produced from the roots of a parent plant. State the name of the type of cell division used to produce new plants by asexual reproduction. This is going to be by mitosis. Then they say, describe why it is an advantage for commercial growers to produce plants by asexual reproduction. Plants produced by asexual reproduction are going to be genetically identical, so we can be able to have the same characteristics of same phenotype without any variation. Also, we can be able to produce large numbers of these plants within a short period of time. So this brings us to the end of question 7. Let's continue to question 8. Question 8. Reflexes are responses that protect the body and involve the central nervous system. Give the name of one part of the central nervous system. This is going to be the brain, or you could say the spinal cord. Then they say, if an animal steps on a sharp object, a reflex act occurs so that it picks up its foot to prevent further damage. Describe how named neurons bring about a withdrawal reflex to protect the animal from damage. Now, a stimulus like stepping on a sharp object is going to be detected by a receptor, and then an impulse is going to be generated. This impulse will travel through the sensory neuron and then to the relay neuron. Neurotransmitters are going to be released as the impulse crosses the synapse, and then the impulse is going to be passed onto the motor neuron, which delivers the impulse to the muscle in order for muscle contraction to occur so that the animal withdraws away from the danger. Moving on. In part B, the photograph shows a breed of dog called a border collie. This is the dog. Some border collies have a genetic condition called sensory neuropathy. Border collies with sensory neuropathy may be injured as their reflexes do not work well. Sensory neuropathy is caused by a recessive allele. They call small n. The dominant allele for not having sensory neuropathy is capital N. The diagram shows a family pedigree for some border collies. So they've given us the key here, and we can see this is unaffected and that is unaffected. This is a female and that is a male. But because these have a child who is affected, then we can conclude that both of them are heterozygous. So capital N small n and capital N small n. Also, these ones have children who are affected, so they're also going to be heterozygous. Capital N small n, capital N small n, and this is small n small n, small n small n. Because these two have a child who is homozygous recessive, it means this one too is going to be heterozygous. So this one here, it's going to be heterozygous. So the next question says, sex determination in dogs is controlled in the same way as in humans. Which combination of sex chromosomes is present in the body cells of a male dog? It's going to be XY because it's the same way as in humans. Next they say, how many individuals in this family have the genotype capital N small n? If we go back here, we have 1, 2, we have 3, 4, 5, as well as 6. So they're going to be 6, and so D is my answer. Then they say, determine the probability that the next offspring produced by individual P and Q is male with sensory neuropathy. The probability of being male is always a half, so probability of sensory neuropathy, we can determine it from here. Because both parents are heterozygous, there is a 1 in 4 chance of producing a child who is homozygous recessive or having the sensory neuropathy condition. So the total probability is going to be that times that, which gave me 1/8. You could write this as a decimal as well. Then down here they say, dog breeders use selective breeding to try to remove harmful alleles from dog breeds. Explain how selective breeding could be used to remove the allele for sensory neuropathy from a population of border collies. So here we could breed dogs with no family history of sensory neuropathy, and then we breed the offsprings of those dogs that do not have sensory neuropathy, and then repeat these selective matings over generations. Or you could determine the genotype and then breed only those without the genetic condition. So this brings us to the end of question 8. Let's continue to question 9. Question 9. The diagram shows the human blood circulatory system, which is this. We can see the head, the lungs, the heart, the liver, intestine, as well as kidney. What is the name of blood vessel labeled Z? Blood vessel Z connecting from the intestine to the liver. That is the hepatic portal vein. And here they say, which labeled blood vessel contains blood with the lowest concentration of carbon dioxide? That should be the blood vessel leaving the lungs, which is blood vessel X. So that is going to be the pulmonary vein. In part B, give two differences in the structure of the blood vessel labeled V and the structure of the blood vessel labeled Y. Let's go back here. Blood vessel V is the vena cava, which is a vein, and blood vessel labeled Y is the aorta, which is an artery. So the vena cava being a vein, it has valves and it has a wider lumen. The aorta has a smaller lumen and it doesn't have valves. The aorta also has more elastic fibers and its walls are thicker in comparison to the vena cava. So I said V is a vein. It contains a larger lumen than Y, which is an artery. V contains valves, while Y does not. V contains fewer elastic fibers, and V contains a thinner wall. Moving on. Here they say, a higher blood pressure and the presence of certain genes are risk factors for coronary heart disease. Give two other risk factors for coronary heart disease. It could be age, it could be gender, lack of activity, obesity, smoking, high salt intake, stress, and so on. Next they say, scientists compared the relationship between blood pressure and the number of deaths from coronary heart disease in Northern Europe, Eastern Europe, and Japan. The graph shows their results. So we can see number of deaths from coronary heart disease per 100,000 on the vertical axis, and here we see blood pressure in arbitrary units. So when we look at the results, we can see for Eastern Europe, the number of deaths decreases until 130 and then increases. For Northern Europe, the number of deaths increase continuously. In Japan, the number of deaths increase continuously, but they are lower than the other two. Moving on. Here they say, a higher blood pressure is considered to be above 130 arbitrary units. Use the graph and your own knowledge to comment on the causes of death due to coronary heart disease in each region. So I said high blood pressure, which is that, leads to increase in the number of deaths in all regions. Here actually I could say above. Let me try to tune it up. Greater than 130 AU leads to increase in the number of deaths in all regions. Japan has the lowest number of deaths at all blood pressures, and this can be due to genetics or any other lifestyle factors. Then Northern Europe had the highest number of deaths. I think you can see it here, Northern Europe. And then the number of deaths in Eastern Europe decreased from 120 AU to 130 AU. The AU is for arbitrary units. So this brings us to the end of question 9. Let's continue to question 10. Question 10. A student investigates factors affecting the rate of photosynthesis. Complete the balanced chemical symbol equation for photosynthesis. We have 6 carbon dioxide, 6 waters, and glucose plus oxygen. Then next they say, the diagram shows apparatus that can be used to measure the rate at which a piece of pondweed produces bubbles of oxygen. Sodium hydrogen carbonate solution is there. This is to provide carbon dioxide. And then we see the pondweed. The lamp is here to provide light, and it's going to be moved in order to vary the light intensity. So they say, the student uses this apparatus to investigate the effect of changing light intensity and carbon dioxide concentration on the rate of photosynthesis in pondweed. This is the student's method. In step 1, fill the test tube with 5% sodium hydrogen carbonate solution. And then in step 2, place a piece of pondweed into the test tube. In step 3, place a lamp 5 centimeters from the test tube. And then in step 4, count the number of bubbles produced by the pondweed in 2 minutes. In step 5, repeat step 1 to 4 with a lamp at distances of 10 centimeters, 15 centimeters, 20 centimeters, and 25 centimeters. That is for varying the light intensity. Then this method is repeated using 10% sodium hydrogen carbonate solution. That means this is going to have a higher concentration of CO2. So here they say, the table shows the results of the investigation. We can see that as the distance of the lamp increases, there is a decrease in the number of bubbles of oxygen produced. The student used the same color of light in the investigation. That means they use the same wavelength of light. So describe how the student could control one other relevant named abiotic factor. They could control temperature using a water bath, or they could control pH using a buffer. Then down here, explain the effect of changing the distance of the lamp on the rate of photosynthesis of the plant weed in both concentrations of carbon dioxide. We know that varying the distance of the lamp is to vary the light intensity. So I said as the distance of the lamp increases, light intensity decreases, so less light is absorbed, so the rate of photosynthesis decreases. In the 5% solution, the maximum number of bubbles is 30 and then levels off, meaning once it reaches 30, no matter how close the light source is, you're still going to get 30 bubbles. While in 10% solution, the maximum number of bubbles is 50 at 5 centimeters, and it doesn't level off. I think you can see it goes until 50. We are not sure if it leveled off or not, meaning if you bring it closer, there is a chance that we're going to see more bubbles or not. So, carbon dioxide concentration does not limit the reaction, but light intensity does. Moving on. Here they say, describe one way the student could modify this method to obtain more accurate results. The student counted the number of bubbles of gas formed, but we know that different bubbles of gas can contain different volume of gas. So if they had used a syringe to collect the volume of gas, the results would be more accurate. Then they say describe how the student could ensure that the results of this investigation are reliable. They should repeat the experiment at similar conditions, calculate the mean, and then be able to identify any anomalous results and make a comment about them. So this brings us to the end of question 10. Let's continue to question 11. Question 11. Biological washing powders contain protease enzymes to digest proteins in clothing stains. Scientists have produced recombinant plasmids containing a gene for a protease enzyme that works at high temperatures. The plasmids have been inserted into bacteria so that the bacteria produce the protease enzyme. Describe how named enzymes are used to produce a recombinant plasmid containing a gene for protease. Named enzymes include restriction enzymes as well as DNA ligase. So we use restriction enzymes to cut a specific gene out of the DNA, and then we use DNA ligase to join the protease gene to a specific plasmid. Moving on. Here they say, design an investigation to find the best temperature for removing protein stains by using washing powder that contains protease enzyme. Include experimental detail in your answer and write in full sentences. I'm going to use this. The first one is conditions for the independent variable. The O is for the organisms. In this case, it's going to be the enzyme. R is for repeat. Then measurement 1, this is for the dependent variable. That is for the specific period of time. And the two things we have to keep standard. So we set up experiments at 5 different temperatures. Then the type of enzyme used, the enzyme concentration, and the concentration of the washing powder should be the same. We should repeat the experiment at each temperature and calculate the mean. Then measure the area or size of the stain after the experiment in order to see the percentage that has decreased. Then carry out the experiment for the same period of time. You could say 1 hour, or you could say for a period of 5 hours. And then the solution should be at a constant pH. It should be at the same agitation strength. I talked about pH because this is an enzyme-controlled reaction, so the pH has to be constant. The agitation strength as well as speed has to be the same because stain removal also requires that agitation. Then the cloth material should be the same. Then ensure that the initial stain is made of the same protein composition and of the same initial size. So this brings us to the end of question 11 as well as to the end of this paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.